Hello everyone, welcome to today's class. So today we are going to learn about the topic that is respiration. We all require energy and we know by the process of respiration we are getting energy. Right? We are eating food, we are doing so many works throughout the day, right? So only eating food will give us the energy. No, it is not going to give us the energy. So what is happening here? So the food that we eat, it is containing various kinds of nutrients. And then they are going to be digested. After that, then they are going to be transported to the various parts of our body, like the glucose. It is transported to different parts of the body after it is getting digested, like the carbohydrates that we are having. Then, in presence of oxygen, it is going to form carbon dioxide. Along with that, it is also going to form energy as well. So, but all of you can see here, ATP is written. What it is? It is basically energy currency of the cell. But what does it mean? ATP means adenosine triphosphate. The full form all of you can write as well. So what should be a good definition of respiration? We can see here that there is breaking of carbon-carbon bond of the complex compound. We know glucose is a 6-carbon compound and we have seen there is carbon-carbon bond. So they are going to be broken. So this process, it is a catabolic process because there is a breakdown happening and it is happening through oxidation that is happening within the cell. That is why it is called as cellular respiration. The students, we require energy. Plants also require energy. But for getting the energy, respiration occurs, we know. And respiration occurs in presence of oxygen and absence of oxygen also. We have been studying it. Aerobic respiration occurs in presence of oxygen and anaerobic respiration occurs in absence of oxygen. So plants, do they require energy? Yes, they also require. But how do they breathe? They also require the oxygen. Yes, they can make their own food. Along with that, they are making energy, that is making the oxygen as well. But there must be something that will be helping them in breathing. We have nose for that. We are inhaling, exhaling easily. But what about the plants? How is the gaseous exchange happens? Does it happen? Do plants breathe? Yes, of course they breathe. So there is little transport of gas from one part to another part. Also, plants do not present, like we haven't seen, they have a great demands for gaseous exchange, like in the root, stem, and the leaves. So we have seen the respiration rates are quite lower than that of the animals or the human beings as well. So what is helping them in this breathing process or gaseous exchange, we can say? Here we can see the stomata is going to help. So the stomata are the fine like pore-like structure that we can see, which are like guarded, or the guard cells are there, which are going to regulate its opening or closing. During photosynthesis, we have seen they are taking in the carbon dioxide and oxygen is released. Now we will be seeing that it is going to take in the oxygen, and after respiration, it is going to take out the carbon dioxide. Similar minute pores are also present in case of the bark or the stem of the plant. They are called as the lenticels. So the stomata and lenticels are helping them in breathing, rather we can say exchange of gases. Okay. So now that we are clear that yes, the respiration is taking place, we have seen one reaction is there. But do you all think that yes, it is only one reaction? No, class, it is not like that. During, we will see this oxidation that is taking place in presence of the oxygen, the glucose is breaking down. All the energy contained in the respiratory substrates are not released freely into the cell in only one reaction. Okay, so we will see there are a series of steps which are there, and that is how we will see that the energy is trapped as a chemical energy in the form of ATP. So we will see that the glucose molecule which is oxidized, don't think that in only one reaction glucose and oxygen they are going to react and form carbon dioxide, water, and the energy. Okay, there are various steps you all could see. There is glycolysis, there is pyruvate oxidation, citric acid cycle, Krebs cycle, which we also call as, and oxidative phosphorylation. Now you all might be thinking, ma'am, we used to know about a simple one-line reaction and now it is becoming so difficult. So many reactions are there. Do not have to worry. I am going to make it simpler for you. In today's class, we are going to focus on the glycolysis first. In the upcoming classes, we are going to focus on link reaction, that is pyruvate oxidation, citric acid cycle, and chemiosmosis. So the glycolysis, or we also call it as the EMP pathway. So why it is called as EMP pathway? Because the scheme of the glycolysis that we can see here, it was given by Gustav Embedel, Otto Meyerhoff, and J. Parnas as well. So what is happening here? So each glucose molecule that we can see here, it is broken down by a series of enzyme catalyzed 10 reaction into two molecules of pyruvic acid. That's great. So where is it happening? It is happening in the cytoplasm, and we know the cytoplasm is present in the eukaryotes, prokaryotes both. So that's great. That means it is happening in all different kinds of living organisms. Yes, it's happening. So no need to worry. And what we can see here, plants, they are making food, right? It is stored in the form of starch, and when it is transported in the form of sucrose. So now the sucrose is converted into glucose and fructose by the enzyme invertase, and then we will see that these two monosaccharide, that is glucose and the fructose, they are going to enter the glycolytic pathway or the glycolysis. So what is going to happen here? Glucose is oxidized to pyruvate, we can see, and the NAD plus, that is reduced to NADH. We are going to see how it is happening, and ADP is going to be phosphorylated into ATP. If you all remember, we know adenosine triphosphate. ADP is basically adenosine diphosphate. Okay, class? So we can see there are total 10 reactions that are present here. Now, out of this, we will see that 1, 2, 3, 4, and 5 are part of the preparatory phase, and the rest are part of the payoff phase. So what are these phases? Let us come to it. So in the preparatory phase, we will see that the glucose, it is going to be forming into glucose 6-phosphate. That means phosphorylation is taking place. And here it is the first priming step also, where we will see in this 6 molecules glucose structure, here in the 6 carbon phosphate is added. That is why glucose 6-phosphate is forming, and it is forming by hexokinase, phosphorylation, and 1 ATP is utilized. So this ATP is used. Okay, here now we can see there is the isomerization reaction taking place, where we see that the glucose 6-phosphate is forming into fructose 6-phosphate. If you can see here, it is a reversible reaction, where in the linear diagram we can see that there is rearrangement from the that is the carbonyl group is going to be transferred from the first carbon to the second carbon. So there is just rearrangement taking place here. That is why it is an isomerization reaction taking place due to phosphohexose isomerase or phosphoglucose isomerase. We can write like this. And then we can see again ATP is going to be utilized. So what is happening? ATP is utilized means phosphorylation is taking place. So here also ATP was utilized. So what is phosphorylation? It means addition of phosphate to a substrate, to a substance or a compound, right? We can see here that the phosphate was added here as well, and again we will see another phosphate is going to be added, and we will see it is going from fructose 1,6-bisphosphate. So now we can see there are 2 phosphate present in fructose 1,6-bisphosphate. So we can see in the preparatory phase, phosphorylation of the glucose into conversion of glucose into glycerol 3-dehyde phosphate. So how the glycerol dehyde phosphate is forming, you must be wondering. And you can see after fructose 1,6-bisphosphate is forming, we will see the aldolase is going to work, and there will be a splitting. And in the splitting, the aldolase is going to work, and this 6-carbon fructose 1,6-bisphosphate is going to be splitting in between the carbon 3 and carbon 4, and it is going to form a 3-carbon ketone. This is a ketone. And we can see there is again a 3-carbon glycerol dehyde 3-phosphate, which we also call as PGAL. Okay, we can also tell it as PGAL, phosphoglyceride 3-phosphate. Okay, so this is an aldehyde. Whenever there is a CHO group, it is aldehyde, and whenever there is a carbonyl group there, it is a ketone, right? You will be learning this in organic chemistry. So once we can see the splitting is happening, you can see here. Now the fifth step, that is the last step of the preparatory phase. So preparatory step is from 1 to 5. Okay, we have studied about 1, 2, 3, 4, out of which you can see class, the first step, it was irreversible, second step was reversible, third step was again irreversible, fourth step was reversible, and we will be seeing that the fifth step, it is also reversible. So we will see that here there is isomerization again happening, and they are both called as triose phosphate. But which triose phosphate will take part in the rest part of the glycolytic pathway? We will see that the glyceraldehyde 3-phosphate, which we also call as PGAL, phosphoglyceride aldehyde, okay, phosphoglyceraldehyde, it is going to take part. So what is going to happen? The glyceraldehyde 3-phosphate directly gets degraded into subsequent steps. But what about the dihydroxyacetone phosphate? Will it not be participating? Yes, it will be also participating. That is why we will see in the rest part of the reaction in the glycolytic pathway, there are 2 molecules of every product are going to form, because once the glyceraldehyde 3-phosphate is going to take part and form the pyruvic acid, then again we will see that the dihydroxyacetone phosphate, by the isomerization using the enzyme triose phosphate isomerase, is going to form into PGAL. So dihydroxyacetone rapidly and reversely converted into glyceraldehyde by the triose phosphate isomerase, and then it takes part in the second phase, that is the payoff phase. Okay, so I hope it is clear, the first 5 steps here. So we can see the glyceraldehyde 3-phosphate is going to take part, and triose phosphate, that is the dihydroxyacetone 3-phosphate, takes part, but it is going to be converted into glyceraldehyde 3-phosphate, then it is going to take part. Okay, now we are going to come to the payoff phase. So in the first step of the payoff phase, we can see oxidation as well as phosphorylation happening. But in this oxidation, what is happening? The glyceraldehyde 3-phosphate that we can see, that is we calling as the PGAL. So here we will see 2 redox equivalent, which are basically the like the H plus present here, that is the 2 hydrogen atoms from the PGAL, that is from the glyceraldehyde 3-phosphate. They are going to be removed and transferred to NADH, and as it is transferred to NADH, it is forming 2 NADH plus H plus, and we will see that the PGAL is also oxidized. So along with the phosphorylation will take place, but oxidation takes place. PGAL is oxidized with inorganic phosphate to get converted into deep, that is BPGA. That means here it is going to be 1,3-bisphosphoglycerate. How it is becoming 2-phosphate? Because here a inorganic phosphate is also there. So the PI that you can see here, class, it is the inorganic phosphate. Okay, so the inorganic phosphate is doing the phosphorylation. Okay, now the 6th step is done. We will see 1,3-bisphosphoglycerate. So 2 phosphates are there. Now, this is a very important step. That is the 7th step. This is the first ATP forming reaction. So here we will see substrate level phosphorylation is done. And by this process, we will see the first ATP is going to be produced. So we can see here that the 2 ADP is taking the phosphate. There are 2 that we can see the phosphate is present in the bisphosphoglycerate, that is 1 and 2 phosphates are present. Now we will see that this phosphate is going to be taken and it is going to take part in the phosphorylation reaction, as this is a substrate and the phosphate group is directly transferred from the substrate or the chemical intermediate to the ATP to form the ATP. That is why it is called substrate level phosphorylation. And we can see that 2 ATPs are produced because twice the reactions are going to take place in the payoff phase. Okay. Now after this, we will see that only 1 phosphate is there. It is going to form 3-phosphoglycerate only in the carbon, 3 carbon carbon number 3, there will be a phosphate. First carbon's phosphate is removed. Now we are going to see in the 8th step there is going to be a mutease working, that is the phosphoglycerate mutease. All the enzyme names are mentioned. All of you can also write it from here. So here we will see just there is a interchange, like from the 3rd carbon the phosphate is coming to the 2nd carbon. That's all, just the rearrangement. Next, we are going to see it is going to remove the H2O. But how the H2O is removed? We can see it here that there is CH2 and we have OH. So what is going to happen? If I draw it here, CH2, CH, and we have the OH, and let's say that's how I'm drawing the 3-PGA, that is phosphoglycerate. What is going to happen? We will see that the OH and this H2 means what? This H2 basically means there are 2 hydrogens present, right? If we draw it like this, so 2 hydrogens are present, right? So here we can see if we remove one H and another OH, it is going to form H2O. So by the removal of the H2O, we are going to see there is going to be a new bond forming. So after the removal of this H and OH, there is a bond in between, and that is how it is going to form as the phosphoenol pyruvate. After the phosphoenol pyruvate, we will see again substrate triple phosphorylation is going to take part. So I am erasing these steps now. How the water is removed, just to show all of you. Don't think that these steps are very like you have to draw it and do it. Not like that. It is just for your explanation class. So we will be seeing that the phosphoenol pyruvate, it is also going to participate in the substrate triple phosphorylation, and the phosphate is present in the 2nd carbon. It is going to like give its phosphate to the ADP, and it is going to form ATP. So again ATP is released. So we can see ATP is released in step 7 and step 8 by the substrate triple phosphorylation, and it is going to form pyruvate. So as the phosphate is removed, so we can see that it is forming carbonyl. So it is forming an acid. So pyruvate, it is also called as pyruvic acid as well. So that is how the 2-pyruvic acid is forming. I hope it is clear to all of you. So let us now summarize what we have discussed. We have studied that glycolysis is happening in 2 phases, preparatory phase and there is payoff phase. In preparatory phase, we can see phosphorylation of the glucose and it is converted into glyceraldehyde 3-phosphate. And we can see that how phosphorylation is taking place and how 2-ATP is used, or we can see utilized in the preparatory phase. Next, if we come here in the payoff phase, we can see 4-ATP is released. Okay, how 4-ATP? We can see first ATP is released in the step 7 and step 10, right? So 4-ATP total. Why twice it is taking place? Because why the whole payoff phase is taking place twice? Because dihydroxyacetone is converting into glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is forming 1-pyruvic acid, and then we will see that dihydroxyacetone is forming another glyceraldehyde 3-phosphate. It is converting into and then forming another pyruvic acid. So that is why we will see in payoff phase there are 2 products are forming all this. Okay, that's why all these are 2 molecules, 2, all these are written. Okay, class. So I hope all of you have understood about it. Now I have a question. That is, what is the net gain of ATP when each molecule of glucose is converted into 2 molecules of pyruvic acid? We can see that one glucose molecule is forming 2 molecules of pyruvic acid, right? Or pyruvate, as I told you. So it is a 6 carbon, and we can see 2 3 carbons are happening. So how many ATP? All right, let me help you in solving. So we can see in step 1 and step 2, there is 2 ATP utilized in the preparatory phase, and next in the next phase we can see 4 ATP is released in the step 7 and step 10. So net gain is our 4 minus 2, that is 2 ATP. So overall we can say in the glycolysis process 2 ATP is like net gain. Along with that, we can also tell that the NADH plus H plus is also occurring. So it is also a net gain, and it will be performing in other parts of the respirations reaction. So after the glucose is produced, we could say it is forming pyruvic acid. So the pyruvic acid formation, it is not requiring any oxygen. That means if it can occur in aerobes, means the organism in which we will see that the presence of oxygen is there, respiration will take place there as well, and we will see that it is also going to occur in the like the organism where fermentation or anaerobic respiration, or in anaerobes it is also going to occur. Okay, pyruvic acid is going to enter by link reaction. We are going to focus on that, how the link reaction is happening. Okay, that means glycolysis is a part of aerobic as well as anaerobic respiration as well. So that's all from my side. In the next class, we are going to further discuss about the other 3 reaction, that's link reaction in between, and Krebs cycle and chemiosmosis, or the oxidative phosphorylation. Till then, everyone, keep on revising and have a nice day. Take care. Thank you for attending today's class. Bye.